Okay, good afternoon, everyone, and thank you for attending this webinar entitled As Told by the Atolls, a narrative from the Tubbataha Scientific Research Expedition 2021. And I hope everyone is stoked, excited, and by the end of my presentation, learn something from it. So for the course of this webinar, as said earlier by Sir Mike, I will be giving a brief overview of the recently concluded expedition of UP Marine Science Institute, introducing the people behind the research, the methods that we used, and some of the research questions that we wanted to answer. And before the session ends, as a promise, I'll introduce to you uh, some wildlife species that we encountered during the expedition. But first, where and what is Tubbataha Reefs? For those who don't know, in the Sulu Sea, Philippines, just east of Palawan, lies an underwater nature reserve that is considered both a mecca for scuba divers and a model for coral reef conservation. So this is Tubbataha Reefs Natural Park, a 97,030-hectare marine protected area and bird sanctuary composed of two huge atolls, namely North Atoll and South Atoll, and a smaller one which is named Jesse Beasley Reef. And within those two huge atolls, there are an islet each. In 1993, UNESCO declared Tubbataha Reefs Natural Park as a World Heritage Site as a unique example of an atoll reef with a very high density of marine species, and the North Islet, located at the North Atoll, serving as a nesting site for birds and marine turtles. In 1999, Ramsar listed Tubbataha as one of the wetlands of international importance. By 2015, Tubbataha was declared by the ASEAN Center for Biodiversity as a 35th Heritage Park. And in 2017, the International Maritime Organization declared Tubbataha as a particularly sensitive sea area that huge ships must be avoided. Being a remote and isolated place, Tubbataha never failed to provide us breathtaking sceneries of serene sunrises, pristine waters, astonishing wildlife, be it on land, air, or deep within its surreal reefs. Our journey began on May 20th, 2021, and lasted for almost 2 weeks aboard the MY Panata, one of UP Marine Science Institute's research vessel, were 15 science crew members of various professions and research interests. There are also 10 ship crew members with us. So allow me to introduce the science crew first, who conducted the research. Uh, Obviously, those aren't our names, rather the different fields of interest that we looked into during the expedition. To understand a complex and large ecosystem like Tubbataha, it's hard to consider just only one aspect. It is better to look at how different facets of the ecosystem interact and affect one another. So, okay, this looks a bit confusing and intimidating, but not to worry. I just wanted to illustrate here how our research questions and interests are interconnected. And for simplicity, I have lumped the researches that are similar in nature and performed by smaller teams of science crew, as denoted by the similarity in color. So let us begin with the first team. The first science crew team is the physical oceanography and the wave glider team, headed by Dr. Cesar Villanoy. We also have Seng Bilguera, Lara Soto, Tin Bantay, me as a volunteer, and we have Jem Barogo and Louis Mojica as inland wave glider pilots. The group aims to study the physical characteristics of the waters surrounding Tubbataha, as well as to track, navigate the wave glider. You might be wondering what is a wave glider. So here is a video of the wave glider, which we named Enrique, named after Enrique de Malaca, the first Filipino who supposedly circumnavigated the globe. This wave glider is actually an unmanned automated vehicle that can take a lot of data from offshore. We use this wave glider to answer the question, what are the physical characteristics of the water far from the atolls? This is a slide showing the parts of the wave glider, showing there are a lot of instruments on it, a camera, a thermometer to measure temperature, a salinometer for measuring salinity, a wave height meter, an acoustic Doppler current profiler for current velocities, and it has also sensors for chlorophyll, A, and refined fuels. It is remotely traced and controlled by on-ground pilots, like I said earlier, by Jem and Louis back in Manila via satellite communications. The solar panel allows charging of the battery so it can go out for months. And as the name suggests, the wave glider harnesses the wave energy in order to propel itself. So as the wave moves up, it pushes the body upward so as the wings. And this allows the glider to utilize less energy from its battery and use the environment to its advantage. To put into perspective how big the wave glider is, here are the photos of its pre-deployment. On the left, we can see Tin, Lara, and Seng preparing the wave glider. And on the right shows Dr. Villanoy together with the other members of the ship assisting for it to be deployed. So here is what it looks like when the wave glider was launched beside the ship. So first, notice how the wings will disappear, these wings. So it descends downwards and then pulls the wave glider, and then it will go off on its own back of the ship and towards where it's destined to go. Okay, so that's the wave glider. Now, allow me to introduce the second, one of the team's anticipated offshore oceanographic events, which was the arrival of solitons. Solitons, as you can see in this satellite image as a sort of a ripple, are kilometer-long solitary internal waves. This actually occurs due to an interference of colder, denser water deep down that interacts with the surface to subsurface warmer waters. So imagine if you have a layer of water and oil. If you tilt the water underneath fast enough, it will create sort of a disturbance in the oil layer. Solitons actually originate on the Pearl Bank, just north of Tawi-Tawi. And the arrival of solitons may introduce colder, nutrient-rich water into the shallow portions such as Tubbataha. This nutrient-rich water may jumpstart primary productivity as it feeds off the phytoplankton in the water. Aside from the wave glider, solitons were observed using a thermistor, similar to the diagram to the left. It is a series of submerged thermometers at a different se- submerged thermometers at a different depths to record if the water gets colder once a soliton pass by. Of course, we're interested to know if solitons affect the Tubbataha reefs. To measure physical characteristics of the waters near the atolls, we deployed on board what we call a CTD unit. CTD stands for conductivity, temperature, and depth. measures conductivity for salinity, temperature, and atmospheric pressure, which relates to depth. For the basic model as shown below, it only has the three sensors, but other auxiliary sensors may be installed, such as what we had on board. Can measure turbidity, photosynthetic active radiation, or the radiation needed by um, phytoplankton to photosynthesize, and of course the dissolved oxygen levels of the water. So here's a video of us with Tin and Seng deploying a CTD from a ship. So after switching on the device, the CTD is automatically turned on and lowered with the help of a winch. It's lowered slowly and will stop at a meter below the surface for 2 minutes to allow the water pump of the CTD to stabilize. After 2 minutes, it is lowered slowly until 200 meters and record. As it descends, it records the parameters as I've mentioned earlier. So now we're done with the first group. We're moving on to the second group, which is the water quality team, headed by the chief scientist on board, Dr. Caroline Haraula. We also have Belay, Rayos del Sol, Lara, Philip Alfonso, Michelle Ranyada, and J.R. Lapresca. For the water quality team, there are two very crucial instruments that they use during the expedition. One of which is a Niskin water sampler, shown on the left. These plastic cylinders are used to take water samples at desired depth without the danger of mixing with water from other depths. Here we'll see Belay, JR, and Lara deployed, how Niskins are deployed. Or sampling beside the ship usually is done with two or more samplers for faster deployment, as seen on the video. The opened samplers were lowered to the desired depth. For the group, it's usually around 5 to 10 meter below the surface. And once it is stable, the messenger, which is what Lara had, is thrown downward, and hopefully it will hit the closing mechanism. For a closer look on how Nissin closes, here is an underwater footage, and focus on the both ends of the sampler. So the Niskin travels along the rope and hits the closing mechanism, and then it triggers the Niskin to close. So so the team is assured that the water that they collected will not mix with the water from the surface or down below. After collection, water samples were immediately stored. Here we'll see Dr. Haraula and JR as they are rinsing the large and smaller PET bottles for storage. So they need to rinse the storage bottles first to avoid contamination. And then on the other side, you can see Belay using a rubber hose to prevent the bubbles from entering the glass jars. So it depends on how precise you'll need, and what chemical parameters that you'll be needing. It may the bubbling might be okay, and then for this particular parameter, the total alkalinity, it matters. So they have to use the rubber hose. And the second crucial instrument for water quality team is the water filtration setup. Here we see Lara preparing the peristaltic pump. She placed a filter on the receiving funnel and then pours the water. So they use the peristaltic pump when they are able to filter. And depending on the type they need, the group may either want the filtrate or the residue from the filter. The two instruments mentioned earlier, the NISKIN and the filtration water setup, applies to the groups of geochemistry, chemical oceanography, and metagenomics. While their methods of data collection are very similar, note that the filter sizes, the volume of water, the sample collected, either filtrate or residue, will be different. Of course, the laboratory analysis will be much different when they land back at MSI. So, for instance, here we have the chemical oceanography group using two kinds of filtering device, the peristaltic pump, as I mentioned earlier, and syringe filters. Their group focuses on the following parameters: inorganic nutrients, chlorophyll A, total alkalinity as a substitute for pH, and dissolved organic matter in order to answer the following question. What are the differences in water chemistry inside and outside the atolls? For Philip, and the sole member of metagenomics team, since he wanted to get the residue in the filter without contamination, he has to flame sterilize the tweezers before getting it. The genetic material of the bacteria is what he's after, and it will be stored in absolute ethanol solution. And the process of metabar coding and shotgun metagenomics seeks to answer what are the differences in microbial composition in water, such as groundwater and different regions of the atoll. Later, I will reintroduce how the groundwater is acquired. For the geochemistry group, uh, members Michelle and Bill, they're interested to know the levels of trace metals, ions in water, nitrate isotopes, and biomarkers in order for them to answer, does Tubbataha experience significant anthropogenic disturbances? Now, another parameter considered in water quality is the presence of fecal indicator bacteria. Water collected from these are directly acquired using a flame sterilized cup instead of the Niskin uh, to avoid a contamination. Here, here's a photo of Michelle serial diluting water samples and mixing it with nutrient growth media. The mixture is then distributed within the wells of quantity tray, example shown on the right. Once the quantity tray is distributed, once the water sample is distributed among the wells of a quantity tray, it is then incubated for 24 to 28 hours. And then when water discoloration occurs, it is marked and will be viewed under a UV light. So those discolored wells that fluoresce indicate the presence of E. coli. The use of IDEX seeks to answer are fecal coliforms present in various waters in Tubbataha. So that's for the second subgroup of the science crew. So moving on to the third one, which I am officially belonging to, is the Reef Assessment Team, together with Dr. Villanoy, Dr. Haraula, Dr. Hazel Arceo, Ravlara, William or Bill Dimalanta, Belay, and yours truly. The, the team's focus was on reef conditions through means of various traditional and advanced novel methods. Traditional methods such as the standard coral reef assessment through fish visual census and photo quadrats require scuba diving. Uh, Using this method, it seeks to answer the question, what are the coral reef conditions and the fish assemblages present in the monitoring sites that were established years ago in Tubbataha? This particular image shows Dr. Villanoy laying a 50-meter transect line at a constant depth, which is a standard opening protocol for these reef assessment surveys. In conducting a photo quadrat method, Bill carries a PVC tetrapod, or what we call an andador, with an underwater camera attached to it. And while he takes photos of the corals, the benthos, for every meter where the transect line is laid, these photographs will be then analyzed in a computer software back here in Manila. So here we see Dr. Arceo conducting fish visual census. Armed only with a slate, she swam along the transect line at a very slow pace, listing down the fish species, the count per species, and estimated sizes that she see in a 5 meter um, 5 meter vision. And those data will be translated later on as fish biomass. So aside from biological surveys that we conducted, sediment samples were also collected by Dr. Haraula, as seen in this video, and on other times by Lara and Belay. These samples are collected by pressing down a 300 milliliter glass jar against the sediment and will be filled by scraping using the the cap as and scraping the additional sediment. And these sediment samples will be analyzed for grain sizes and lipid analysis to answer if there any influx of nutrients and sediment depositing that is originating from the land. So those are the traditional methods that we used for the reef assessment survey. Let's move on to the more advanced methods, starting with hydrophones. So these hydrophones are actually underwater sound recorders that in the photo it is being retrieved by Dr. Arceo and Dr. Villanoy. These underwater sound recorders were left on for 12 hours overnight, and they wanted to find out if they can detect underwater biological choruses that are influenced by animal behavior, and then later on relate it to how good or bad the coral reef condition is. The team also used BRUVs. It is short for Baited Remote Underwater Video Stations. These stationary seafloor camera stations use bait to attract fish in the vicinity, and it records the species attracted to the bait or just passing by the camera lens. Here we'll see Dr. Arceo, Lara, and Dr. Villanoy deploying a unit of BRUVs. So the unit is just thrown over the boat, and with a rope they assist the descent of the whole camera system, and will be left underwater for 45 minutes to an hour. Using the BROVs, they can answer the question, what are the predatory fishes present in the area that are not detected from FBC? Since large fishes tend to avoid a sight of humans, so they might be underrepresented in the fish visual census. So I don't have the exact footages taken during the survey, but here is an example of what how it looks like when it's recorded. So notice that sharks and other carnivorous fishes are attracted to the bait. Okay, moving on to a more familiar instrument since I was the one operated this is the Rapid Assessment Instrument for Coastal Benthic Habitat System, or Arecobe system. What I'm holding in the video right now is actually the camera component of the system. which I will deploy right now. So the camera is just lowered on the side of the boat. The video on the right just shows the how it looks like underwater. And once the camera is was deployed, the boat is the camera is then towed by the boat, moving from shallower portions to deeper parts of the reef. It is set on time lapse and taking images like these. And with it, I can answer the question, what are the coral reef conditions around and inside the atolls? Since this is a boat-based survey, I was able to cover more area for a short period of time. Aside from the camera, Araikobe system is also composed of a single beam echo sounder, as shown by it is attached to the PVC pipe, as shown holding by Seng here, and as well as a GPS to record the location. And my surveys won't be a success without the assistance of my fellow STEM workers, Philip, Tin, Seng, Lara, Mary, JR, and Michelle, who operated the two other instruments while I manned the camera. So last but not the least, the research sub teams is the ILET team, together with Dr. John Birkin Leong, Dr. Haraula, and Mary Limen. Uh, they explored they explored the soil composition, configuration of the islets, and explore the possibility of presence of fresh water within those islets. Well, speaking of fresh water, allow me to introduce the first instrument they use, which is the piezometer. Piezometer, which is currently being hammered down by Dr. Ong, are long, hollow metal tubes that allow groundwater to surface. So once the piezometer was buried deep beneath the soil, a rubber tube with a hand pump attached to the other side was fed into it. This allows to draw water on a faster rate. Okay, here we'll see Dr. Haraula using the pump to extract groundwater. So as you can see, there is groundwater present in the islet, and these groundwaters will be tested for electrical conductivity in order to determine how close the groundwater from being freshwater. Also, as mentioned earlier, these samples that collected using the piezometer will be tested for metagenomics and IDEX by the water quality team. Another possible way to determine presence of groundwater is subsurface electrical resistivity. So Dr. Ong and Mary laid down these electrical wires with steel rods across different portions of the islet, and connected to it is a electrical switch box which injects low direct current. Since different materials have different electrical resistivity, for instance, freshwater has higher resistivity compared to a soil or rock, it can translate a rough sketch of the underground, such as this image. So the more yellow the graph, the more possible that it is freshwater. Using electrical resistivity, it can answer the question, what are the potential composition of the subsurface material within Bird Islet? So again, this is just potential. It's not the actual. For the actual subsurface composition, the team actually used a sediment corer. So a 2-meter sediment corer was hammered down using with the assistance of a steel weight. So here we'll see how the core is buried deep within the soil. And then the weight is reeled up in the metal frames and released directly above the core. And when it reaches the 2 meter mark, the core is then reeled upwards with the help of a chain block. This is reeled slowly so uh, the the core itself would be prevent in crumbling, and the integrity of the core will not be compromised. So here are Dr. Haraula and Dr. Ong with the help of marine rangers unveiling a portion of the sediment core. So aside from the metal portion of the core, they have the acrylic portion which cases the sample. So notice the differences in the color of the acquired core sample. It has a darker brown color from the left side, which is the surface, and as it goes deeper or going to the right portion of the core, it turns lighter. It goes white. These samples will also be subjected to grain size and lipid analysis, similar to the sediments acquired in scuba diving. The islet team also wanted to answer the question how permeable the soil is in different sections of the bird islet. Here we see Dr. Haraula with volunteer Seng and Tin drilling a hole using a cylindrical shovel in preparation for the permeameter. So this is a portion. This is a cylindrical shovel that is made for this expedition. So this is the actual permeameter as being held by Dr. Ong. This is a 3D printed water container with a measuring tape on its rectangular cuboid end here, and a porous filter on its cylindrical end here, allowing the water to escape. It is inverted and inserted directly in the drilled hole. So they're just waiting for it to stabilize. So after the water level is stabilized, observers like Belay and Philip will note the initial water level. So as you can see, they're pointing where the water is stabilized, and they will note the subsequent water level after a minute. And essentially, the method measures the rate of water escaping from the container into the earth. So the slower the rate of change, the less um, permeable the soil is. So last but not the least for the islet team, the islets are crucial to be monitored for any signs of changes in its shape due to sea level rise. One of the cost-effective method employed was shoreline tracing using a GPS. So Mary in this image shows and together with other marine park rangers, they walk along the coast of the two islets while holding the GPS and creating waypoints for every 10 steps. These waypoints were then plotted on a map using a GIS software. Another method is aerial photography using a drone. So after taking aerial shots, it will be then compared with the aerial shots taken from the previous years. So now that I've mentioned all the teams, research teams, allow me to introduce the rest of the expedition crew, starting with the last member of the science crew, which is Ivan Serenas. He was the man behind the scenes taking those stunning photos and videos of us in action. And later on, you'll see more of his uh, captures for the wildlife portion. Of course, I won't forget also to introduce the hardworking members of the ship, headed by the Captain Edgardo Cruz. and Chief Mate Abraham Kindoy, Second Officer Rayan, um, Kuya Ricky, Kuya Rolando, Kuya Reynald, who mans the maintenance of the boat, Brother Mike, Brother Kuya Jeffrey, and Kuya Bagtong, our boatmen, and Kuya John Ray, who cooks our delicious meals during the expedition. We also had on board Cheche and Chuchay, our dog crewmates. And throughout the course of the expedition, from planning and execution, the UP Marine Science team was assisted by the Tubbataha Management Office and park rangers. Their team, led by Protected Area Superintendent Angelique Songko, regularly conducts research in Tubbataha in order to better understand the ecosystem and protect the Tubbataha from threats. So when I say threats, threats from what exactly? So one of the few threats we saw during the expedition was the presence of marine debris. These plastics, judging from their appearance of being discolored, it looks very old, and most likely be drifted by currents, or in the case of the photo to the left, carried by seabirds as materials for nets. So these marine debris are also observed by the Tubbataha Management Office, albeit very few in volume, since the Tubbataha Rangers regularly clean the park. These marine debris still pose danger for life. As shown here, there was a ribbon stuck in the shark. Unfortunately, they cannot go closer to the shark to set it free. And as presented earlier, changes might occur in the islets due to erosion caused by severe storms and sea level rise due to climate change. Since 2016, Tubbataha Management Office is on alert with the possible endangerment of the resident seabirds once the bird islet is gone. Global warming also affects the corals of Tubbataha, with prolonged exposure to um, elevated seawater temperatures. The corals expel the symbiotic algae that provides not only the color but also nutrients, making them more susceptible to death. So now that I've given you the rundown of the people and the science, we'll move on to the second half of my session. One of the major reasons why we need to understand Tubbataha and protect it. I also wanted to emphasize that this is no way a very exhaustive list of the biodiversity Tubbataha has to offer, meaning these are only just very few of the animals that we saw. So Tubbataha Reefs Natural Park is a home for no less than 360 species of corals, or about half of the all coral species in the world. So corals are actually animals that are closely related to sea jellies and sea anemones. They build up calcium carbonate skeleton over time, and they build different. They build it with different forms. So here's a picture of corals with different forms. We have the tabulate form, and we have a branching that goes upward and outward. We have branching form that has thicker branches. We also have here the massive type. This is a Porites coral that builds corals in a hemispherical shape. And of course, the branching, as I mentioned earlier, this is a mixture of Acropora, the green ones and the brown ones. This lighter brown one is a Porites branching, and the thicker branching here, as I'm pointing, is the Isopora. We have also the Porites massive on the right hand side. Also, I also saw one of very big colony of Acropora, which is a tabulate form. This tabulate form is around 4 meters in length, if I remembered correctly. So these four life forms of Acropora really extends extensively. And moving on, there are also 600 species of fishes recorded in Tubbataha. So here we are seeing the damselfish. Looks like they're feeding on plankton. So just jumping off the boat, no, we already saw a lot of fishes. For example, the trevallies or the jacks or talakitok. So here are some of the still images that I was able to took when they're not very quickly swimming away. And here are the trevallies forming a school. Moving on, there are also we've also found surgeon fishes. Surgeon fishes are characterized by these blade-like structure uh, near their tail. So these are very sharp, like a scalpel. So hence why they're called surgeon fish. We also found unicorn fishes near the surface. Unicorn fish because of the pointy end on their heads. So Ivan was lucky to encounter and video a battery of barracuda. So these schooling barracudas, I think these we estimated it to be more than 1,000 individuals. So these barracudas are very long silver fish with stripes on it, and they are very aggressive when they're not in school form. So here they're very calm because they are not hungry. Moving closer to corals, we have reef fishes like we saw like the Moorish idol, and we also saw trigger fishes. So these are the black fishes that uses the that flaps their fins up and down. For a closer look, here's what triggerfishes looks like. So they use these fins to primarily to move, and they have very sharp teeth as well. We were also uh, saw butterfly fishes. Butterfly fishes are actually good coral indicator. So if you found a coral reef that has a few or more species of butterfly fish, then it might it indicates that it might have a very good coral reef health. Okay, for this one, I hope you can see the grouper slightly camouflaging and resting on a bed of soft coral. I haven't found the grouper. The grouper is this one. And for my favorite fish, the parrotfish, uh, they are named parrotfish because of the beak-like structure that they have to use to scrape microalgae in dead coral, live coral, and in other sediments. And we also found the largest species of parrotfish, which is the bumphead parrotfish. As you can see, they have a hardened structure in their forehead. So they use these hardened structure to break down corals and other surfaces so they can feed on the microalgae. Moving on, Tubbataha was also known to have 11 species of sharks and rays. What we have here, actually, we're still debating if this is a white-tip shark or a gray reef. It looks like a white-tip shark. So here are some of the still photos we acquired for other sharks. Unfortunately, since we cannot go closer to them, we cannot identify them successfully. However, we were lucky to encounter the biggest fish in the world, whale shark, just swimming past by the ship. So some of us also encountered rays, such as stingray, like what you're seeing right now. Other rays that the other team members saw were eagle rays. Unfortunately, we haven't encountered any manta ray, as other the park rangers said that encountered after we left the Tubbataha. So the bird islet and south islet are also home for 100 species of birds. So some of the species of birds that we saw are the brown booby. So they have these plumage of brown, almost brown throughout their body, while they're young, start with very white feathers. We also saw red-footed booby, characterized by, well, their the color red, with their webbed feet colored red, and they have the whiter plumage. We also saw brown noddy. These bird species have their nests on grasses. There are also great crested tern that lay their eggs on the sand. Um and we also have the sooty tern, similar to the great crested tern. They lay eggs on the ground, on the sand. And we also saw the only breeding pair of masked booby. So there's a story behind this. Around 1990s, they were thought to be extinct in Tubbataha, but then they in until 2016, when marine park rangers uh, found them, and hopefully they success they are successful in bringing back the population of masked booby. So aside from birds that lay their eggs in the islets, there are also nesting green and hawksbill turtles. So here are the still images of green sea turtles that are just resting on coral surfaces, and this sea turtle is actually being followed by this species of batfish. Uh, Here are other images of um, the sea turtle just resting. So here is a video of the hawksbill sea turtle. It was captured swimming and eventually taking a rest. Okay, so here's the hawksbill just taking a rest inside the crevasse. And last but not the least, for the wildlife gallery, Tubbataha also have recorded 13 species of dolphins and whales. So in this video, we can see what it looks like a bottlenose dolphin that are swimming along the MY Panata. So I'm not sure if the banging of the ship really actually attracts these dolphins, but looks like it. See, the another dolphin just comes closer and then swims away. This is another pod of dolphin with another species, which we think are striped dolphin, due to them having a longer snout and having those striped formation. So again, these dolphins just swim beside the and in front the uh, research vessel that were aboard. So for the final video, we were lucky to have a close encounter with the same pod of dolphins as I um, featured earlier. Here are us riding a rubber boat with the dolphins with our genuine reaction. So watch out kasi may sound, maraming shouting na magaganap. So enjoy. And with that remark, I think that perfectly wraps up the entire experience of the trip. Before I formally end my presentation, I would like to thank the following: the Commission on Higher Education, and Philippine California Advanced Research Institute, or CHED-PICARI, for funding the research expedition, UP Marine Science Institute for making it happen, Tubbataha Management Office led by Miss Angelique M. Songko, the park rangers, Philippine Coast Guard, and Navy who assisted us during the expedition, MY Panata crew, Dr. Caroline Haraula, the chief scientist, for allowing me to share the knowledge, Dr. Villanoy, Dr. Arceo, and Dr. Ong for parting us with their knowledge as well. And last but not the least, my fellow STEM workers, Lara, Belay, JR, Michelle, Bill, Mary, Seng, Tin, Philip, and Ivan, for a very wonderful and memorable experience. And thank you.